Hi everybody, we're going to pick up with the muscular system and the sliding filament theory of muscle contraction. In your course pack, this information begins on the bottom of page 84 and continues to the top of page 85. So let's start by reviewing the myofilaments. You may recall there's two types of myofilaments, thin filaments or actin filaments, and thick filaments made up of myosin. On this picture, we're going to ignore the elastic filaments. Which of these two filaments is anchored to the Z-line? Well, that is the thin filament. It is anchored to the Z-line or Z-disc. The thick filaments have the myosin heads, these little knobby protrusions that you can see sticking up here. Let's also review the banding. We notice there's two types of bands, an A band and an I band. Those A bands are the dark bands composed of thick filaments and what else? Thin filaments. Okay, I bands are, of course, the lighter of the two bands. They are composed only of thin filaments and Z-lines. Let's also talk about the H zone. It's a special portion inside the A band. What's missing from the H zone? No thin filaments. And of course, probably most important here is the sarcomere. That is the distance between what? That's the distance between two Z-lines. That is the unit of the myofibril that we notice will shorten during muscle contraction. Let's also review the specifics of each of our filaments. The thick filament is made up of myosin. That's the protein that composes it. Those myosin heads are protrusions that stick off the surface of those myosin filaments. Actin is actually more than just actin. Actin filaments contain troponin and tropomyosin. So I jokingly say that you can remember them as AT and T. So actin or thin filaments include a rope-like tropomyosin, which covers binding sites on the thin filaments. That binding site is important because it's where myosin will bind when muscle contraction takes place. What is the role of the troponin complex? Well, this troponin complex binds calcium and it regulates the exposure of those binding sites on the actin filaments. So it's important that we appreciate the role here of troponin being a regulatory protein. It regulates when that tropomyosin rope can move away and reveal those binding sites on the actin filaments. Alright, so let's walk through the steps. They begin on the bottom of page 84 and continue on to the top of 85. When a nerve impulse known as an action potential arrives at the muscle cell, it travels into the sarcoplasm and through T-tubules, which are indentations of the sarcolemma. That way, the action potential or nerve impulse can reach these myofibrils. Recall that myofibrils were composed of these actin filaments and these thick filaments known as myosin filaments. That action potential causes the sarcoplasmic reticulum to release calcium. When that stored calcium floods out of the sarcoplasmic reticulum, the calcium travels into the sarcoplasm of the muscle cell. It binds with this troponin complex here, and when that binding occurs, tropomyosin moves. Then it reveals binding sites on those actin filaments. Here you can see what happens when troponin and calcium have been bound together. Those ropes of tropomyosin moved out of the way, and now we can see the binding sites available on the actin filaments. We call them myosin binding sites. That is where the myosin head will reach up and perform an action known as a cross bridge. Here you can see cross bridge formation has taken place. That's when this myosin head here will reach up and attach to the binding site on these thin filaments. After cross-bridge formation occurs, a power stroke will occur. That allows the myosin head to shift and pull that thin filament. So the head binds here and it moves in this direction. That causes this entire thin filament to shift and be pulled. So in effect, the myosin heads are causing the thin filaments to slide past the thick ones. That is the foundation for the sliding filament theory. In order for this myosin head to release and repeat the process, we must power this process with ATP. So ATP lets that head release from that binding site, relax back down into what they call a cocked position. It's kind of like a snake ready to bite, And so in just a moment, that head can reach up and form a binding site and repeat the process once again. So what we're watching here is a repetitive process. The head reaches up, binds, forms a cross bridge, pivots, makes a power stroke, pulls that thin filament, and then ATP powers the release. All of this requires for calcium to be bound to that troponin complex sitting on the actin filament. Now, if there is no ATP available, then that myosin head becomes adhered to that thin filament and unable to relax. When this happens, people are usually dead, and that's causing rigor mortis. It occurs a certain number of hours after death, and then the body begins to degrade. So within 24 to say around 72 hours post-mortem, the stiffness associated with a dead body tends to go away. Now, this picture is kind of busy, but the myosin filaments are shown here in red. And this, of course, is an actin filament. And you can see numerous myosin heads reaching up and forming cross bridges and power strokes. This process allows for the thin filaments to be pulled and slid past the thick filaments. So the filaments here do not shorten themselves. They only perform an action that causes them to slide past one another. That is the basis for the sliding filament theory. Myosin heads binding, forming cross bridges, pivoting in the form of a power stroke, and then pulling and sliding those thin filaments. When those thin filaments slide past one another, we move Z lines closer to Z lines, and that causes the entire sarcomere to shorten. So in this image, what you're watching is a myosin head walk along, forming cross bridges and power strokes. The myosin head is shown as the red golf club, and of course the actin filament has a blue rope of tropomyosin, a yellow set of actin filaments, and then the troponin complex. What is the molecule that must bind the troponin complex in order for the rope of tropomyosin to move out of the way? Well, that's calcium. Watch very closely for that yellow calcium to bind to that troponin complex. Now, in order for that head, just having performed a power stroke, to release from the actin filaments, we must power it with the release of ATP. So ATP is split into ADP, adenosine diphosphate, and an inorganic phosphate molecule. So that process of having the head release so it can repeat is driven by ATP. So keep in mind, when the head binds to the actin filament, you see the cross bridge. The head pivots. That causes a sliding effect with the thin filament. That is a power stroke. This repetitive process will cause the muscle to shorten as the heads pull Z lines closer to the center of the sarcomere. In effect, the sarcomere will shorten, not the A bands, of course, and not the thin or thick filaments themselves, but the I band disappears, and of course that H zone disappears as well. So here you've got a look at the sliding filament theory just by looking at the bare bones. This is, of course, a relaxed muscle, and how do you know that? Well, there's a very distinct and visible I band. The I bands are only visible when you have a relaxed muscle. As the muscle begins to contract, we assume that those myosin heads are anchoring to those thin filaments and walking and pulling along those thin filaments. As muscle contraction continues, we find that the I bands completely disappear, as does the H zone. All right, so take a close look there. That H zone should have been through the middle of this A band. It is gone. I bands are gone. They've disappeared. And if you watch the distance between the Z lines, that would be our sarcomere. And of course, the sarcomere shortens as we see the muscle contract. So sarcomere shortening is our goal here. So this is basically pictures 1, 2, and 3. And let's identify what we know here. Here is a Z line. They've got it labeled right there. Here's another Z line. Watch those Z lines as this muscle contracts. Watch how they get closer and closer together. Also, watch for the H zone. The H zone begins to disappear. In fact, it's not labeled in the bottom two pictures. So when the sarcomere contracts, the sarcomere shortens. Okay, that's one thing. Another thing that happens are I bands disappear. What else disappears? H zone disappears. Okay? What else happens to this thing? Well, Z lines move how? They move closer together. We really don't talk about the A band there because it doesn't really change length. It's that sarcomere and the length between those Z lines that really changes length here.